Welcome to all the participants after the tea break for the second session by Dr. Raghukumar on Mycelium, the world of fungi. When I came to Goa in 1996, whenever I used to hear about the scientists, I always used to hear four names, and those four names were Dr. Unthavle, late Dr. Unthavle, then Dr. Raghukumar, Mrs. Dr. Raghukumar, Lata Raghukumar from NIO, and the fourth name was Professor DJ Bhatt. So every time, whenever mycology was heard about, all these four names, and Dr. Unthavle was working on the mangroves. So every time I used to hear these names, but I never ever got the opportunity to interact with Mr. and Mrs. Raghukumar. But today I'm fortunate enough to have Dr. Raghukumar with us. And so it's my honor and privilege to introduce Dr. Raghukumar, an internationally acknowledged expert on biology and biotechnology of marine fungi. Presently, he is the Chief Scientific Officer, Evisa Myco, San Francisco, California, and a Managing Director of Mycotech Private Limited. He has received his master's degree from Madras University, and he did his PhD on marine fungi under the guidance of famous mycologist Professor C.V. Subramaniam at the Center for Advanced Studies in Botany, Madras University. He worked as a scientist at the Institute for Marine Research, Bremerhaven, Germany, between 1976 and 1981, during which time he was groomed by Dr. Alvin Gredner in the taxonomy and ecology of the marine fungi belonging to Throstocritris. He also worked as a scientist from 1982 to 2004 at NIO, Goa. His major research interests at NIO included the biology of marine protists, the Throstocritris, biomass of fungi and Throstocritris in marine samples, the ecology and taxonomy of marine wood degrading fungi, microbiology of marine detritus, the microbiology of the Arabian Sea and marine biotechnology. He participated in numerous oceanographic cruises and headed several research projects during his tenure at NIO. In addition, he has taught several courses in marine microbiology, biodiversity, and biotechnology. Sir has published more than 80 highly cited research papers in international and national journals and has also described several fungi which are new to science. Presently, he holds Indian and US patents in the area of marine biotechnology. He has been a visiting scientist at various institutions abroad and was the last outgoing president of Mycology Society of India. With this brief introduction, on behalf of Department of Botany, I welcome Dr. Raghukumar and I request our TY student to welcome him with the floral bouquet. I now request Dr. Raghukumar to take over the session. Thank you.
Thank you. Good morning. Yeah, 10 more minutes for morning. Okay. You're all graduate students. I don't know how much of fungi you know. Fortunately, Professor Bhatt gave you a very fascinating introduction to you about fungi. And I'm going to keep to the basics because I don't know how much fungi you know about. Probably in school you read a little bit about mucor, same in your, probably a little bit about Aspergillus, Mycelium, Penicillin, etc. now in your BSc courses. So, but the idea of my talk is to talk about something very fundamental to fungi, not go too much into the depths of fungi, but talk about something very fundamental. because I would like you to appreciate the basics of any biological system around you. For example, every organism has something special. When you talk of trees, they grow up in height. The birds, avians, they occupy the air. So each group of organisms has something fundamental, something special. And understanding that in biology, whatever group it may be plants, it may be fish, it may be birds, whatever you like, appreciating that around you, I believe, is what is most important. You may continue with fungi, you may continue with something else, but the lesson that you should take home is that you need to keep your eyes open and appreciate the basics of biological organisms around you. So when it comes to fungi, where is next year? Okay, yeah, I'm good. Yeah. So, okay, both bacteria and fungi do the same job, right? Both have an absorptive nutrition, both are osmotrophs, they're heterotrophic, osmotrophic heterotrophs. Both do the same thing. So, what do you think then is special about fungi? Okay. In any ecosystem, no two organisms can do the same thing and still survive, right? There is always a competition. Anything which does the job better will overtake and then suppress the other one. That's the basic principle of ecology. For example, if there are two hairdressers in the same area, there is going to be a lot of competition, and one of them probably will have to be doing the ladies' hairdressing much better, the other gents, something like that. So each organism, each group has something special. So between bacteria and fungi, what do you think is special about fungi? Okay. Anyway, the answer was already in the title of my talk. So the unique thing about fungi, of course, is a mycelium, right? And this is just a cartoon of a fungal mycelium. Look at the beautiful structure. Sometimes when I see a picture like this, I wonder how nice it would have been for the city planners to take an example from fungi for a developing city and plan according to it. Probably people are thinking about such a thing, but something starting from here, let us say a small village, a small town, expanding in terms of the road system, connectivity, transport, and things like that. So this is what is the mycelium is the most fundamental thing for fungi. I would like to go a little bit into how this fungal mycelium came into existence. You need to know a little bit about what happened in the past. The past is always very fascinating, the evolution. For example, if you go back in time, life on land, you see the trees here, you see the sediment, you see the birds, you see the animals. You are talking about wildlife, tigers, whatever, a mouse. All that never existed till about 600 million years ago. There was hardly any life on land, right? And life on land started probably somewhere around 600 million years ago. And today is somewhere here, the Holocene, somewhere around the Proterozoic, from somewhere around the Cambrian, Precambrian is when probably life started developing on land. The fungi most logically evolved in the sea because fungi and animals have a common ancestor. And the multicellular animals had already developed in the sea. The sponges were the first multicellular organisms. They are marine. And if both of them had a common ancestor, the fungi should have evolved in the sea. But there's really, unfortunately, no evidence for that, except for protein sequence clocks. People use protein sequence changes, what are called as molecular clocks, to understand the possible time when any group of organisms could have evolved. And these protein sequences analysis provides sort of evidence that fungi were there around 1,000 million years ago. But there was no life on land, right? So, although there is evidence that fungi evolved about 1,000 billion years ago, the actual fungi on land came about probably about 600 million years ago. If you see this is a sort of a phylogenetic, the evolutionary tree stating where the fungi originated. This group is the fungi, and that is the root of the fungi, and that is shown as somewhere around 1,400 million years ago, right? The vascular plants are here somewhere about 900 million years ago. So this is what the evidence suggests. But life on land, as I said, did not evolve till about 600 million years ago. Most probably, the evidence suggests that life on land came because of a symbiosis between a phototrophic organism and a fungus. This happened about 600 million years ago. The present understanding is that some freshwater phototroph, something related to charophycy, was the first organism that came onto the land along with the fungus. And this partnership of fungus and plant has changed the entire atmosphere, climate, the sediment formation, life on land, and everything that exists now. The only true evidence that we have is through fossil evidence. There is fossil evidence by about 600 million years ago of the first plant-fungus symbiosis in the form of what are called as arbuscular mycorrhizae. These are fungi which reside inside plants. These are the arbucles. There are fungal mycelia here. These are all from inside a plant. So the first symbiotic association probably arose about 600 million years ago between where the fungi were in the form of an arbuscular mycorrhizae belonging to a group called Glomeromycota, as we call them today. And that seems to be the first evidence of fungi and plant association and on land. And as W. and Stewart very nicely put, we can state unequivocally that without green land plants, there would be no land animals, including humans. But I can say add to this, without green plants and fungi, because the evidence is that both of them came onto the land at the same time. There are many symbiotic associations. One is, of course, the lichens. You all know about lichens. These grow on rocks. And when you take a lichen and take a cross-section, you will see fungal mycelium over there and the algal zone. Together, they have a symbiotic role. These kind of symbiotic associations seem to be the very first land organisms that evolved. There again is a picture of the same. Here is the fungal mycelium here, the algal layer. As I said, probably some plant belonging to the charophyceae came onto the land along with the fungal association. And what happens in such an association is when you have bare rock and you have an association like that, there is all this association of a plant and a fungus starts causing erosion of the rock. And in those days, at this time, these are presumed fossil plants, Aglaophyton, Horneophyton, with an arbuscular mycorrhizae inside, as we can see here. At that time, the sediment was probably 1 millimeter to 2 millimeters. That's all there was sediment there. There was nothing else there. It was rock, some amount of erosion. So that's all was there. So with further plant fungus associations, symbiotic associations, and more erosion, such as this, the mycorrhizae. This was the arbuscular, these mycorrhizae are inside the plant, and then you have ectomycorrhizae. All these kind of plant fungus associations, in all likelihood, were responsible for the formation of land or pedogenesis. The implication of that is that it is very important even to the marine environment, because the erosion is what caused the leaching out of silicates from the rocks on land into the sea. We all talk about diatoms in the sea, okay? You probably have heard about diatoms. These are the most important primary producers in the sea. There are no diatoms till about 200 million years ago, because there was hardly any silicate. All the silicates came from land because of the erosion that was caused, in all likelihood, between plant and fungal association. So gradually the soil developed, and so now presently you have all the life there is on earth thanks to this association. So the importance of fungal mycelium for life on land is very, very fundamental. No life on earth without the mycelium. Now, what are the special features of a mycelium? Of course, I talked about the mycorrhizae. You know about we all see in many cases mushrooms above the ground. Very rarely do we know that these mushrooms actually have a symbiotic association in the form of fine fungal mycelium, the ectomycorrhizae, that connects the mycelium to the tree. Okay, the mushroom is actually the end product of this ectomycorrhizal mycelium, which has finally fruited and come above the soil to give you the mushroom. So this is, I talked about the very early arbuscular mycorrhizae within the plant, a lichen-like symbiosis, and ectomycorrhizae, 3 different levels of fungus plant associations, which are all important in pedogenesis. Here is a picture to tell you, look at this is all root, of course, interval. Look at this fine fuzz here, the ectomycorrhizae. Practically every plant that you find has an ectomycorrhizal connection. They say there is hardly any plant you can count which plants do not have a mycorrhizae rather than which plants have a mycorrhizae. So this is the kind of an association which has been fundamental on Earth and which prevails now and provides all the benefits to your plants that you see around you. You see the trees, you see the plants, but you do not realize that it all comes from the fungal mycelium that is there under the ground. Connect this to I have a couple of videos. If this is connected to the they are connected to the net, right? Two more. Okay, yeah, there you go. Okay, one of the okay. One important thing I think the basic thing about the mycelium that I find is that it is capable of flat horizontal growth on any substrate. It does not need free water. Bacteria need free water to move around and multiply. The advantage of the fungus is that it can spread horizontally. For example, if you take any fungal growth, any fungal colony, the growth of it, If you inoculate a bacterium and a fungus on a prickly plate, the difference would be this, like this video is showing you. Okay, you stick a bacterium on a plate here, you get like this, okay? It's all over there. Okay, but it has all stayed in the place. It happens very fast, but it's not spreading horizontally on the plate. But take this fungal culture here. In no time, the fungal colony expands and covers the entire plate. Look at the colony here. There. Look at the size. And in no time, it covers the entire plate. A bacterial inoculum, when you put on a plate, it will stay at that place or just where your streak, around the streak. Whereas even when you do a spot inoculum of a fungus in a plate, it spreads horizontally. This is one of the fundamental characteristics of Mycelium. Thank you. I love it. Both of you. Okay. So this is one of the fundamental characters of the Mycelium. It is horizontal spread. Now, this is common. You all know this. You have seen that. But have you ever appreciated that, for example, during damp season, anywhere, particularly a place like Goa along the coast, west coast, with the damp walls, you get a covering of fungus, right? Everything is covered with fungus, particularly black fungi, right? So this is the capability of the Mycelium. This is one of the fundamental characteristics of the Mycelium is its capability to spread horizontally without the help of free water. All that it needs is a moist surface and humidity. That's all that needs. This is a common sight for you again. This is once again, you have seen this. Do you recognize this? This is called a sooty mole on leaves. This is a mango leaf. Very common. Everyone who has seen a mango tree around you, you can recognize that. Now, this again has no free water, just a moist surface, and the fungus has spread horizontally all over the entire surface. This, as I said, is a fundamental characteristic of the fungus. Now, in both the cases, let us say a fungus is growing on a damp wall, somewhere outside on the compound wall, or on a mango leaf. What is the stress factor there, other than, okay, it has happily started growing, but it is exposed to one danger. What is the danger? Can you tell me what is the danger? Anyone? No. The sunlight. The sunlight is a danger because the sunlight has the strong UV light of sunlight is harmful. That is the reason why these fungi that I showed you have a black color. They are called Dhempe fungi, black fungi. They have the pigment melanin in them. I will also come to another example. This is again a very interesting case. This is a fungus growing on the. You might have heard of a nuclear disaster in Russia at a place called Chernobyl. There was a lot of background gamma radiation, deadly gamma radiation. If you and I stood there without any protective clothing, we'll probably gone in 1 week. But people notice that there are fungi growing there on the walls of the Chernobyl reactor in spite of the heavy gamma radiation. Again, these were all had the melanin pigment in their mycelium. So when a fungus might be able to grow outdoors on a damp wall or on mango leaves, or in this case on the Chernobyl reactor, what it needs is a pigment melanin. The melanin is an excellent absorbing compound that can absorb UV light excellently. For example, this graph shows you the wavelength of light starting from UV at 200 nanometers to red at 700. And this is the absorbance of the melanin. And it has got a strong absorbance all over till 700. Particularly at this very high energy UVC light at 200 and then up to 400. This entire region is UV. So the capability of melanin to absorb UV light is one important reason why the fungal mycelium, in spite of being exposed to open air and to the sun, is able to protect itself to survive. Because UV light generates a lot of dangerous reactive oxygen species like hydroxyl radical, hydroxide ion, singlet oxygen. All these are very, very dangerous to health. These can disrupt many of the biomolecules within the organism. For example, in human beings, what is the danger of UV light in human beings? Come on, please, guess. What is the danger of UV light? Sorry? Right, okay. I thought you were going to say tanning. Tanning is not the danger. Tanning is a good thing. Although people like to use Fair and Lovely. Tanning is a good thing. Skin cancer, especially for Caucasians, is a very, very dangerous thing. So UV light has all these problems. Dimer formation, telomeres alteration of proteins, lipid peroxidation, inflammation, all this leading to skin cancer. And it can, similar damages to the cell can happens also in any organism, including fungi. So UV light can cause a lot of damage to DNA. And that's the reason when there is melanin there, which absorbs UV light, the skin or any cell, a biological cell is protected. So there is a reason why these fungi, fungal mycelium, exposed to sunlight or gamma radiation have melanin in them. So that is what is common for all the fungi as well as human beings of all races and colors. Bird feathers have melanin. Did you ever know that bird feathers have melanin actually? The various colors are actually caused by different configurations of melanin nanoparticles. We are not talking about that in detail now. The cuttlefish sepia, when in danger, it oozes out an ink, defensive ink, the black ink, that is also melanin. Many black insects have melanin. So here is a fungal colony which has melanin. So melanin has a fundamental role in protecting the mycelia when they are exposed to UV light. These are some of the fungi which are famous for having melanin. This is called alternaria. This is called curvularia. These are just fungal colonies. I think these are the yeast hortea. Now, what happens in the fungal cell wall is these are the nanoparticles of melanin arranged in layers, and there's a cell wall, there's a capsule in the cell wall. This kind of an arrangement is what is believed to particularly protect against gamma radiation. Gamma radiation is a very deadly radiation. Cancer patients are given a very, very low dose of gamma radiation as a radiation therapy. But these fungi apparently tolerate even much, much higher doses of radiation thanks to the melanin pigment that is present in the mycelia. Talking again about horizontal growth. If you walk through the forest, you will see these rhizomorphs, right, on the ground. Especially in monsoon season, it's fascinating to see them. Thick strands of mycelium, thick strands of hyphae, which are rope-like and very visible. I'm sure most of you or all of you have seen this. Other important feature is you take a Petri plate with inoculate and mucor and presume you have got on top of the lid, again you have some medium there, coated with medium. Within a week, the top also will start getting the growth of mucor because fungi have the mycelium. An important advantage is that it can traverse space. It can grow through empty space, starting from a base with nutrients. It can grow into empty space and till it reaches another substrate and then grow there again. So capture of space is what I call it through aerial hyphae is a very important feature of mycelium. These are all special features which fungi possess and which enable them to survive in the ecosystem. Okay, the forest floor is actually a very thick interwoven matrix composed of the mycelium. You hardly ever know that there is so much of fungus inside when you just walk on the soil, but there is a whole lot of mycelium that is there, an intricate web of fungal hyphae. Another important feature of mycelium is this. You see, Professor Butt talked about Ganoderma today. You see this on logs, wood, decaying wood. What you see is only the end product. Fungal mycelium is characterized by a very strong apical growth, right? Because of its apical growth, the apical growth works this way. You put a certain pressure, you take a needle and put a certain amount of pressure. The needle will penetrate. But you put the same amount of pressure on a flat body and press it, nothing will happen, right? The pressure at a tip, fine tip, helps the filament or the tip to penetrate into a substrate. The additional advantage being also simultaneously it secretes enzymes. So this fundamental property of an apical growth helps the fungus penetrate into substrates. So there might be a lot of bacterium outside. This is one very interesting technique I always try to do in my lab when my fungus is contaminated with bacteria. There is a bacterium fungus growing together. I don't even very often have to add an antibiotic. You have the fungal, let's say inoculum on the bottom of the plate and put a layer of agar over that and then leave it like that. The fungus will penetrate through it and come out. The bacteria most often remains inside. There is one way, easy way that works very often for fungi. So this property of apical growth penetrating through a solid surface, this is a sketch of a tip of a hypha. It is very rich in calcium, called the Spitzkoerper, this entire structure. So that is the reason if you cut a section across, let us say wood, you will find a dense web of mycelium inside. They are penetrated, right? They are there. They are living there till the time is favorable for them to produce a fruit body outside. So we talked about a horizontal growth. We talked about the capture of space. We talked about the penetration of the hyphae, their capability to penetrate. These are all important properties. These are all again, this again is a wood vessel of a wood where you can see the fungal mycelium. Okay. But then the mycelium has to be formed from somewhere, right? It's apparent that the means of forming a fungal mycelium is from its pores. So most of these fungi which produce mycelium and which produce a fruit body outside are dispersed through air. I need your help again. Oh, okay, you're doing that way, right? That I'll do next time. Okay. Sorry. I've got two more like this. Sure. No problem. You can do it. They try to brainwash me through these apps. Okay. Not there. Okay. No, this is not the one. This is not the one. This is the previous one. I think you have pasted the same thing again here.
Okay. Just close it.
I think the earlier one was there.
I'll just use it. I closed the new one. I think I missed it. Sorry.
Oh, yeah, there it is. Okay. I just find this thing very fascinating. This is very, you can see this also in Goan forest. It's called a puffball, right? Again, these Mycelium, which where the fruiting body has come out of the soil, and then it's called a puffball because the spores, earlier they used to have the powder puffs. So when you press it, the spores come out like a puff. So it's called a puffball. Look at that. This actually, this is from the people who made the movie, The Fascinating Fungi, right? There is a movie, you should watch that. Okay, anyway, there you go. But if ever you do that, I suggest you always wear a mask. I wouldn't do that. you can never predict. Even if it were an edible mushroom, that's a different thing. But the spores are a different ball game altogether. Okay, yeah, that's it. You're all different from what. We don't need that. I'll go back to my presentation. It's okay. I'll close that. I'll close that and go to. I can
No, don't close it. Close my.
Okay.
Wait.
Okay. Now, I have been so far talking about mycelium on land, but we have been most of the time we talked about the conquest of land by the fungi, right? Somewhere, some ancestral fungus came onto the land because of its symbiotic association with plant. But looks like many of the fungi have gone back to the sea from the land, right? So you find fungi also in the sea. These are fungi that have gone back. For example, particularly if you see in mangroves, Goa is rich in mangroves. The whole stretch is really, Ashish has done a lot of work for us at Mycotech, collecting a lot of Seshagiri and we had some very good ore from many mangroves in Goa. Now, again, if you take the mangrove wood, the same thing happens. The mangrove wood is perpetrated inside with fungal mycelium. This is one wood fiber. See the mycelium going through it, a diamond-shaped cavity is caused by cellulase enzyme, and then it narrows down, penetrates fine, then again creates a diamond-shaped cavity because of cellulase. So this wood fiber is permeated by the fungal life right inside and getting its fruit from the cellulose of the fiber. And if you take a mangrove wood and slice through it, you'll find fungal life inside. And having done that, the fungal mycelium is right inside. As I said, thanks to the apical growth and penetration. And then outside, they will produce their fruit bodies. These are aquatic fungi. marine or mangrove fungi or mangelicorous fungi. And the evidence is that fungi migrated from land to the sea. And you will see food bodies and a variety of beautiful spores, ascospores. The characteristics of the ascospores is that, yeah, look at them. These are all sticky appendages. Help them, they float in water and then they stick to a surface and germinate. This is very typical of aquatic fungi. Or they have radiating these kind of appendages which help them to keep afloat, right? Because once the surface area is more, it tends to float better than less surface area. So, and they have sticky appendages. So a variety of beautiful appendages and spores. Dr. Damodar Shana is an expert on mangrove fungi. He has done a lot of work, continues to do so. He knows a lot about this group of organisms than I do. Look at that. These are all a variety of beautiful mangrove fungi. That would be Peritrichospora, Damodar? No. Oh, and that is Halosarphia, I suppose. That is Halosarphia. It is Halospheria tricornuta or something. Okay. Okay. Now, all that is about the advantages of mycelium. But there is also a disadvantage. The disadvantage is this. You take a flask with medium and take a small bit of fungal inoculum and put it there. On the other hand, you take a drop of a bacterial inoculum and put it in the cultured medium. In 24 hours, the bacteria have penetrated, spread all over the flask medium, turned it turbid, right? That is the advantage of a single cell. If you have taken a fungal inoculum and put it in a flask, it will stay there, stay put there, struggling to grow slowly, right? It's very difficult for fungi to grow totally immersed in seawater, unless, of course, in fermenters, when we grow them, we give them, agitate them so that there is the air, there is a mass transfer of air. So that's what we do. But in general, it is the bacteria that have an advantage when there is free water. So what organisms do is, when they have a problem like that, they try to overcome that problem by adapting to a different life strategy. Okay, so for example, this is typical. Bacteria are unicellular, fungi or mycelial. And as I said, each one has its advantage and disadvantage. You cannot get a bacterial coating or a damp cell wall the way you get a fungal growth, right? And you can't expect a bacterium to traverse the air and touch the top of the Petri plate. You can't expect a bacteria to grow on a plate as rapidly as a fungus if you put a spot in it. But then, as I said, the advantage is when there is free water available, bacteria have that advantage, fungi do not. So in trying to adapt to a different life strategy, many fungi are turned to a unicellular form, and which you know. And these are the yeast. Okay, so these have more or less the same advantage as bacteria. And on the other hand, there are bacteria which have adopted a mycelial form, and these are the actinobacteria, right? So there is also a difference in the lifestyle strategy depending on the situation. The fungus doesn't have to be always mycelial. A bacterium doesn't have to be always unicellular. There you are. All your breweries all depend on your Saccharomyces cerevisiae. Now, another big advantage of a mycelial form is this. We grow when we want a metabolic product, any bioactive compound, any commercially useful product. We grow them in bioreactors, in fermenters of huge sizes, 10,000 liters, 20,000 liters. We do that also for fungi where it's a liquid medium and you can cultivate the fungus with proper aeration so it grows. Bacteria grow very effortlessly in a liquid medium, there is no issue. So, but the advantage is the mycelial form is you can grow them in another form in what is called as a solid state fermentation. Right? So the substrate is just damp. There is no free water there. So you inoculate your fungal inoculum. Since fungi can grow on a moist surface and spread very easily through it, and if you take a substrate like that, that might be air grabs, go through those, spread very nicely, and so there you have a very good solid state fermentation. Your mushroom cultivation is based on solid state fermentation. There are many commercial products formed through solid state fermentation. So that is another advantages of mycelial form. Mycelial fungi lend themselves now to many, many biotechnological applications. You take a mycelial mat, grow a mycelial mat, and there are companies now which are trying to make myco leather, right? So no more, they don't want animal skin. It's going to be more expensive than animal skin, but there is a tendency to now look at myco leather. There are companies that have started making this. It's very attractive. They have the same property as leather, quite tough. Fungal mycelium is made of chitin. It's a very tough molecule. So, and the fibers are tough, and you are able to make things like myco leather or myco bricks. People are now mixing fungal mycelia with other matrix materials and trying to make mycelial bricks. Sorry.
Sure, sir. No problem.
This is something I wanted to show you before I concluded my lecture. But before that, there is another very interesting product made with fungal mycelium. This is a fungal mycoprotein. There's a product in the market. Many people are looking at making mycelial proteins. The one that is in the market is called corn, Quorn. You might have heard of that. It is made with the fungus called Fusarium nivale, and it is in the market. In the US, at least, it is very. I suppose I don't know about the UK, but the technology was developed in the UK. But I have had an occasion to eat corn in the US, where there are different kinds of product of corn available. For example, like a beef in texture or taste. So for vegetarians who don't like beef or whatever, then my friend who is a vegetarian, strict vegetarian, wanted to have a taste of beef. And then he said, okay, let me buy some corn and eat. So single cell proteins, particularly mycelial proteins, are now becoming a very important aspect of fungal biotechnology. This last slide, I just was going through the net and I found this a very interesting slide. Fungal mycelia are multinucleate, right? And they are heterokaryotic in the sense the nuclei are genetically different. So you don't, they're not, the other thing is homokaryotic. So the heterokaryotic fungal nuclei, there are so many of them in the fungal mycelium. They move around in the mycelium. And this particular video is a fascinating video showing how the nuclei move around in the mycelium. And the only is
It's full.
okay, that's maximum. I've stopped here because when the video is going on, you will see how this nuclei, when it comes to to this place, the septum in the hypha, they squeeze through the center. Just watch that. They squeeze through. So that's the fungal mycelium for you. So, if you liked it. It's an amazing structure. You just say mycelium, but there are various ways of looking at mycelium. Whether you go into a forest soil, whether you slightly dig the soil and see the fungal mat under the ground, or whether you even look at the fungus spreading on a wet wall. Yeah, all those are fascinating things as far as mycelium is concerned. And yeah, so keep your eyes open and enjoy the fungal world around you, and as well as I like to call myself a biologist because there are many aspects to a biology of every organism. And I'm most familiar with fungi, but at the same time, there are a lot to look around and admire, including the fungal mycelium. Thank you.